Hello, everyone. I'm Tiff Boltwood, the Managing Director of Australian Genomics, and it's my very great pleasure to be co-hosting this webinar today on global research inequity and opportunities for childhood dementia with the lovely Kim Hemsley.
Thanks very much, Tiff. Yes, my name's Kim Hemsley. I'm the head of the Childhood Dementia Research Group here at Flinders University in South Australia, and it's very exciting to be here today and talking about such an important topic.
We'd like to begin by acknowledging the traditional custodians of country throughout the lands we're joining from today. I'm on the lands of the Wurundjeri Woiwurrung people of the Kulin Nation, and I'd like to pay my respects to elders past and present and extend that respect to all First Nations peoples joining us for the webinar today.
I think today we're going to have a presentation of the results of the research and followed by a panel discussion. Then to ask questions, if you can please type them in the question and answer box. We'll take some questions in the last 20 minutes of the webinar. And the session is being recorded and it will be shared after the seminar is over. Okay, I think I can start by introducing the first three of our panelists. I'd like to introduce you to Felicity McManus. Felicity, thank you very, very much for being here today. Felicity is a member of the Family Advocates Program at the Childhood Dementia Initiative, and she's based in Victoria. She's mum to 3 children, and her eldest, Orla, is aged 12, and Orla has alpha-melanocytosis, a disorder that causes childhood dementia. I'd also like to introduce you to Dr. Matthew Ellinwood, a long-term colleague. Matthew, very kindly joining us from Durham in North Carolina in the United States. He is presently the Chief Scientific Officer at the National MPS Society, where he does research to benefit the patient community. Prior to that, Matthew has more than 2 decades of research experience. He was Professor Emeritus at Iowa State University, where he carried out basic and applied research on neuropathic MPS disorders, including developing and testing multiple treatments, including enzyme replacement therapy and gene therapy. In his present role at the MPS Society, he is a fierce advocate for multiple initiatives, including the implementation of newborn screening and regulatory approval policy. Thanks very much, Matthew. A pleasure to have you here today. And finally for me, Dr. Leslie Meltzer. Thank you very, very much, Leslie, for beaming in from Boston in Massachusetts in the US. Leslie is a Chief Medical Officer at Orchard Therapeutics. In her role there, she manages clinical development, regulatory, medical affairs, diagnostics, and patient advocacy, and oversees the regulatory strategy and supports the commercialization of OTL-200, which is a recent FDA-approved treatment for early onset medical leukodystrophy, a childhood dementia-causing disorder. Leslie leads the expansion of newborn screening for MLD, drives early and late-stage clinical development of the company's next in-line gene therapies, and these are the treatment of Hurler and Sanford-based syndromes. Thank you very much, Leslie.
And it's my privilege to introduce our final 2 panellists. Professor John Christodoulou is based at the Murdoch Children's Research Institute in Melbourne, where he's the director of the genetics research theme and co-leads the brain and mitochondrial research group. John also holds the chair of genomic medicine, the Department of Paediatrics, University of Melbourne. In addition to mitochondrial disorders, John's major research interests are in Rett syndrome and related neurodevelopmental disorders. He also has a major research interest in the application of next-generation sequencing technologies in rare genetic disorders. Welcome, John. And finally, Dr. Chris Elvidge, who is a stranger to no one on this call, I imagine. Chris is the head of research at the Childhood Dementia Initiative. She's been working in rare disease patient organisations since 2008, including 5 years at San Filippo Children's Foundation, and has been at CDI since its launch in 2020. She's passionate about accelerating the development of much-needed treatments and completed a PhD in molecular biology at the University of Western Australia and a postdoc at the University of Oxford. Chris, over to you for the presentation.
Thanks, Tiff, for the introduction. So I am going to give you a brief overview of the results of the landscape analysis that we recently completed. I'm not going to go into a lot of detail and leave a lot of the discussion for the panel. But if you would like to see more detail, you can find the full reports on our website. So for those of you who are not familiar with childhood dementia and perhaps missed our previous webinar, childhood dementia is caused by more than 145 rare genetic conditions. And when you bring all of these causes together, it affects 1 in 2,900 births. And sadly, the median life expectancy is just 9 years of age. And these statistics are from a paper that was published in the journal Brain towards the end of last year. So in this landscape analysis, we focused on clinical trials because for the vast majority of children with dementia, clinical trials are the only opportunity to access a potential treatment that has any hope of changing their disease course. So what we did was looked for all the clinical trials for all the types of childhood dementia that have been registered in clinicaltrials.gov since that database started in 2000. And we found 386 clinical trials for analysis. Next, we compared the level of clinical trial activity to that for childhood cancer. So why did we do that? So not everyone knows this, but childhood cancer is a group of rare conditions. That's what childhood dementia is too. Both childhood cancer and dementia are serious and devastating for families. You might be surprised to hear that both groups of conditions cause a similar number of deaths each year in high-income countries. So in Australia, where I'm based, childhood dementia is estimated to cause about 91 deaths per year, and childhood cancer causes 92 deaths per year. And this is replicated across the world. So they're the figures for the UK and for the USA. So what did we find? We found a real inequity that spanned over the full 23 years of data available. And what does it look like currently? So this graph shows the number of clinical trials per child with dementia and per child with cancer. And we found that there were 24 times more trials recruiting for childhood cancer than for childhood dementia. Of course, we want to see more childhood cancer research, and we celebrate the advances that are being made for childhood cancer. And to be clear, we don't want to see less childhood cancer research. We just want to see more childhood dementia research. And also beyond this inequity, we found some concerning trends, and that research for childhood dementia seems to be slowing down. This graph shows the number of new childhood dementia clinical trials started each year. And with the scale on the right there, in the light blue line is for childhood dementia, reaching a peak around 2020, 2021 of 30 trials initiated. And since 2015 onwards, the number of new trials seems to have plateaued. And there may be many contributing factors to this, including the pandemic and the global economic downturn. But you can see that the number of trials registered for all diseases, which is the dark blue line, has continued to increase. This dark blue line has a different scale on the left-hand side there. And with all of the technical advances in genomics and drug development, we would expect and hope that the number of clinical trials for child dementia would be increasing exponentially right now. And unfortunately, the story only gets worse. We scanned recent press releases and found worrying evidence of many trials being halted despite positive results and no safety concerns. We found 10 examples of this. that are listed in the report. There are likely other examples, but it's difficult to identify them. 9 of these trials were still listed as active in clinicaltrials.gov at the time of analysis. So the results that I presented on those last couple of trials, a couple of slides, actually overestimates the clinical trial activity for childhood dementia. And we found another trial, 5 trials that failed to initiate at a late stage. For example, after natural history studies had been completed, drug manufactured, and toxicology studies completed. Reasons for these failures include regulatory and commercial barriers. We've heard about companies needing to run longer and longer trials, being denied access to the accelerated approval pathways, and regulators demanding unfeasible clinical trial designs. This makes trials more expensive, and ultimately companies, especially small biotechs, can't continue. And the end result? Promising treatments are shelved, which is outrageous. It's ethically wrong, heartbreaking for families, and incredibly wasteful of decades of research, mostly funded by patient organisations and governments. Okay, changing tack a little here, I'm going to turn our attention to the Australian context because a good proportion of the audience today is based in Australia. Of the 54 clinical trials recruiting patients globally in December last year, only 2 had sites in Australia. And due to the narrow recruitment criteria for those 2 particular trials, that means that less than 2% of children with dementia in Australia would be eligible for those trials. And by contrast, in Australia, every child diagnosed with cancer is eligible for at least 1 clinical trial. And we also noticed that no new trials opened in Australia in 2023. We also looked at the research funding allocated by the Australian government to research. And since 2017, there's been almost 5 times more funding allocated to childhood cancer. It's anticipated that this lack of research funding for childhood dementia is replicated around the world due to the lack of awareness of this group of conditions, which have traditionally been seen as individually rare conditions rather than grouped together based on their similar clinical presentation. This historic and ongoing lack of research funding is contributing to a global lack of clinical trials and subsequently extremely limited treatment options for children with dementia. There are opportunities all the way along the drug development pipeline to address these barriers that I've talked about and to increase the availability of effective treatments for children with dementia. So this diagram shows the drug development pipeline, right from basic research to understand the cause and disease process of these conditions, drug discovery and optimization, preclinical studies to get ready for clinical trials, then clinical trials, which can be multiple phases, and then finally the regulatory approval and reimbursement process, which makes the drugs available to patients. And as I've said, we need to increase research funding, which will feed the clinical trial pipeline. We need to improve the commercial viability and clinical trial efficiency so that we don't see these late-stage failures, and to encourage more pharma companies to enter this field. And we need to really streamline the regulatory approval and reimbursement process, so which will feed back into improving the viability of clinical trials. And all along this pipeline, we need to increase awareness of the unmet need and the unique characteristics of this group of diseases. And, I just go back, I missed a point. There has been a lot of talk recently, particularly about this, the regulatory approval process in the US with the FDA, and there was an excellent workshop held by the Reagan-Udall Foundation last month, which discussed some of these barriers, and we hope that progress will continue to be made in this area. And we'll talk a bit more about that in the discussion, I'm sure. Continuing on the topic of hope for change, this is a graph from the Swedish Childhood Cancer Foundation, but the pattern is replicated in high-income countries around the world, showing impressive gains in survival over the past 50 years for childhood cancer. In Australia, for all childhood cancer, the survival rate is now well over 80%, but there's been no such improvement in survival for childhood dementia. If the data were available for childhood dementia, it would be barely visible along that 0% line at the bottom. We'd like to be able to show a graph with improvements like this for childhood dementia in the near future. So starting to wrap up now, I just wanted to bring the therapeutic research into context. I'm sharing the Venn diagram that we use to give an overview of the work we do at CDI. For anyone who's unfamiliar with our work, you can see how all of our areas of activity are interlinked. Research needs to be embedded in improvements in care and support systems. For example, if we don't have the care systems for early diagnosis, we won't be able to find the patients early enough for emerging treatments to be effective. All of our work converges on getting treatments and cures, equitable and quality care, and making childhood dementia a health policy priority. And finally, the families directly affected by childhood dementia are at the centre of all the work that we do. So I'm finishing with a photo of little Mia, who had childhood dementia caused by Batten disease and died at 9 years of age. I want to read to you something that her mum said. So she said, what would Mia's life have looked like if she was born 15 years from now? Will it be different? Or will it be a different story for the next generation of children yet to be diagnosed? Or will things be at a standstill? Thank you for the families who share their stories and contribute their voice and experiences to our work. Finally, I'd like to acknowledge our Scientific and Medical Advisory Committee and everybody at Childhood Dementia Initiative for supporting this work, especially our fearless leader, Megan Mack. And I'll hand back to Kim and Tiff now for the panel discussion.
Excellent. Great. We're all here. Fantastic. I think, initially we might kick off just particularly coming from Chris's final slides with Mia and Peter's comments. Felicity, I might start with you. Thank you for joining us today to share, a very personal story. I understand your daughter Orla had a bone marrow transplant, which is an intervention that's only applicable to a small number of children with dementia. So of the 145 different conditions that cause childhood dementia, only 6 can potentially be treated by bone marrow transplant. And even then, only some children, if diagnosed early enough and a donor match is found. Can you talk about your family's experience with accessing treatment for Orla, and what do you wish people better understood about the urgent need for new therapies? Oh, you're on mute, Felicity. All right, we might, Felicity, if we could see if you could work out your technical issues, I might pass to Kim to ask another question.
Sure. Thanks for that, Tiff. Leslie, if I can maybe move to you now. It's absolutely fantastic to hear about the recent FDA approval of Orchard's gene-modified cell therapy. I understand it took a very long time, 15 years, and I hope you can address that. But I'm wondering if you can please tell us a little bit about how the therapy works and also what the results have shown.
Sure, Kim, and thanks so much for having me today on this panel about a very important topic, very close to my heart for the past few years. So the therapy that we've developed in collaboration with our research partners at the hospital in Milan, Italy, is for metachromatic leukodystrophy, which is a disorder in the list of childhood dementias. It's a lysosomal storage disease and a leukodystrophy. And the approach that the therapy takes is actually built on the backbone of the bone marrow transplant that Felicity was mentioning, which is only an option for a very small number of disorders. And the approach takes the patient's own stem cells and genetically modifies them to correct the genetic disorder that's seen in metachromatic leukodystrophy, which allows the bone marrow cells to engraft in the body, migrate to the tissues and differentiate into cells that are important in the tissues and express very high levels of the enzyme that's missing or absent in patients with the disorder. So we have an ability to biochemically correct the underlying genetic defect in metachromatic leukodystrophy. This is a disorder where children typically will develop normally and then in the most severe form will plateau and rapidly lose their motor abilities, their cognitive abilities, and it is life-limiting. The majority of children will die within 5 years of disease onset, unfortunately. And this therapy, through many years of research in over 37 patients who were treated at that single site in Milan, has demonstrated benefit on slowing or even preventing certain manifestations of the disease. It also showed a survival benefit in children with the most severe form who were treated. At the age of 6, 100% of children treated with the therapy had survived, whereas only 58% of children who were untreated had survived by that same age. So it's been really wonderful to see the benefit. There are side effects associated with it, as it is with any therapy. It is currently approved in Europe, where it's been approved for a few years now, and it was recently approved here in the US, and we're very excited about the opportunity to bring the therapy to patients in those two geographies and around the world in the future as well. But yes, I think it was a longer journey when we acquired the program. Back in 2018, all of the work, like I mentioned, had been done in Europe. And so we had to initiate those discussions with the FDA from the very beginning after the study had already been going on for many years. And I think a critical element in making sure the FDA really understood the severity of the disease and the potential benefit that the therapy was offering was that voice of the patient community. The patient community really came together and had numerous educational sessions with the FDA about the disease, about what happens when it's not treated, and about the children who'd received treatment. And in our interactions with the agency, they frequently cited how impactful those interactions were with the patient community. So I think it really speaks to the patient voice in helping regulators understand the severity of these diseases and the opportunity that having a therapy like this can create for these children.
So important. Thank you, Leslie.
Sure.
And back to you, Tip.
Thank you. And speaking of the patient voice, I hope Felicity will be able to join us again to share her story. And as much as I'm accused of loving the sound of my own voice, that wasn't a very pleasant experience for all of us. So apologies to attendees. Matthew, I might pass to you. As a researcher and now a patient advocate, why do you think there have been so few clinical trials for childhood dementia disorders?
I think overall there have been too few clinical trials, but I think if you dig down into the numbers, there have been an embarrassment of riches in some disorders and a wasteland, as it were, in others. And part of that is our understanding of the diseases and also the underlying metabolic defects in the diseases may make it particularly challenging in one case and particularly more attractive in another. I work in enzymatic lysosomal storage diseases. The concept is pretty clear. If you can get the enzyme to the patient's cells, you can see a resolution of signs. So those are fairly simple. As it were, at least from a theoretical standpoint. Other childhood dementias, they have a more complex cell-based etiology, and just figuring out the basic science about how to treat them can be more demanding. Even though, however, when we have cases where theoretically the path towards treatment is more straightforward, we still have serious roadblocks or hurdles when it comes to the regulatory process, enrolling trials, structuring trials, and a lot of that has to do with the innate complexity of the disease. Our diseases are effectively developmental disorders on top of neurodegenerative disorders. And those are the two groups of diseases that are probably the most difficult to come to a treatment for clinically. In the case of neurodegenerative diseases, we're not there even in the age-related neurodegenerative diseases. At least in the childhood dementias, we know that there's some underlying metabolic or genetic defect. It's figuring out how to treat those. But once disease pathology begins, once that cascade of disease has started, it can be difficult to intervene. And so that comes into another issue, which is making sure that we get to patients early enough. And how is that managed when our diagnosis is based on clinical presentation? We do have the technology to do newborn screening, but there is a long-standing tradition that you do not screen for these diseases until you have treatments. The consensus around that may be changing because we do know how to help manage the diseases in ways that we did not in the past. Furthermore, advances in being able to do high throughput targeted genomic sequencing of newborns may help identify these patients at an earlier age. That could help enroll clinical trials. Their potential response to therapy at a younger age may be more beneficial. So I think there are a lot of potential solutions on the horizon, but they're not here yet, and they're not here fast enough for our patient community.
Thank you, Matthew. Felicity, speaking, Matthew's comments about different interventions available, can we try to hear your story now?
Yes, sorry about that, Tiff. I think, can everyone hear me now?
Yes.
Yeah. So, Ola, my daughter, was diagnosed at 16 months, which is quite a very young age to be diagnosed, but she was showing symptoms of the condition, which is obviously how she came to be diagnosed. I suppose what we didn't probably quite understand was how severe she possibly was. So we were quickly put in touch with the cancer team at the Children's Hospital, and were very fortunate to get a match from an unknown donor within 3 months, and she was in the bone marrow transplant 3 months later after diagnosis. At the time, we were put in touch with a family advocate in New Zealand who had 2 adult children with alpha-melanocytosis, and he then consequently put us in touch with a couple of families in the UK who had children that had had bone marrow transplants a bit older than Ola. So from my understanding at the time, she was the youngest child transplanted in the world, I think. And that was really helpful to talk to them. We did scoured for a lot of information as much as we could. I mean, even at the time, a Google search didn't give us many pages of information. And although it was helpful to talk to the families, it probably didn't really prepare us for what was to come, because every child's so different, and that's probably what we were lacking in information really too. The transplant itself was very high risk. We were told that there was a 1 in 10 chance that she wouldn't survive. And when that can seem like low risk, I suppose, but when you've been diagnosed with a 1 in a million condition, it's pretty high risk. And although she came through the transplant okay, it was pretty touch and go a few times. So it was a pretty traumatic process to go through. We were also, at the time, there was a company overseas who had a clinical trial in very early stages, proof of concept, and they did offer to enroll her in a trial, but that was probably even higher risk than the transplant, because we at least knew that if she survived the transplant, it would have some impact And yeah, the actual bone marrow transplant itself was very finessed, I suppose. They really understood what they were doing, but they didn't know anything about how it would really help her. So it was, we were jumping into the unknown, essentially. And even now we're still facing, I suppose, risky, unknown procedures, because she still needs to have ongoing orthopedic surgeries, which we have no data about that as well. So it's the ongoing care as well is very well, not very well understood, I suppose, too.
Which is, I mean, incredibly hard, as I imagine, as parents navigating that uncertainty, you know, even with clinical carers, you know, the lack of determined pipelines and support must be really challenging.
Yeah, it is. It is. And yeah, I mean, most often the thing that you turn to is information and research and people that have done it before. And when you're the ones that are the first to do it, it's pretty hard. Yeah.
And hence the importance of your ongoing advocacy. So, you know, thank you so much for joining us today and sharing that, Felicity.
That's okay.
And maybe a question for you, John. You know, clearly Orla, Felicity and their family have had limited recourse to therapies in Australia. I'm wondering if you can maybe address how we might be able to get more clinical trials into Australia and available for the range of families that we have here with childhood dementia conditions.
Thanks, Kim. I guess picking up initially on some of the points that Leslie and Matthew made, issues that are relevant, you know, regardless of where the clinical trial is going to be taking place, you know, to be able to introduce precision therapies, it's essential, obviously, to have a genetic diagnosis. And so that can be an initial challenge that families face from the outset. And, you know, in some instances, the therapy is gene variant specific. So, for instance, allele-specific oligonucleotides. So having a precise genetic diagnosis is essential. And then for the sort of rare and ultra-rare disorders, apart from having a good understanding of the underlying biology, the next challenge is to have a deeper understanding of the natural history of the disorder, because that's essential for being able to identify clinically relevant outcome measures, you know, be they clinician reported or participant reported, and may help in identifying relevant laboratory-based outcome measures that could be incorporated into the clinical trial process. So they're sort of general issues. If we look at the Australian context, given their very nature, in terms of being very rare, it's unlikely that any one site will be able to recruit enough individuals from their own jurisdiction to be able to make the establishment of a clinical trial a viable option. And so ways to potentially deal with that are, firstly, partnership with patient support groups is incredibly critical in terms of making sure that the communication and messaging and all of that is taking place. And then the other point there is that establishment of a rare disease clinical trial centre of excellence is a complex process, and you need not only individuals who are experienced in running potentially complex trials, but also clinical experts in the disorder who are going to help inform things. And so a potential model in Australia would be to establish a network of paediatric clinical trial centres where you could draw in the appropriate skilled force that would allow sharing of experience and expertise, and that would maximize the ability to run clinical trials efficiently. And I guess ideally, because these things aren't cheap, it would be great if we could convince federal and state governments to partner to provide some infrastructure support for setting up these networks. And I think that would make it potentially much more attractive to bring clinical trials in if it was a partnership in that regard, in partnership with pharma companies. And I guess the last point I'd make, picking up on something that Chris flagged, was there could be really important lessons that can be learnt from our pediatric cancer colleagues, who have been really successful in brokering access of their patients into clinical trials. And they've really done a great job in being able to sort of seamlessly link the clinical work with the clinical research. So they're the sort of initial thoughts I have.
Chris, would you like to expand on that? I mean, certainly from your presentation, the inequities in research investment in clinical trial access compared to other rare conditions, is stark. And the report certainly, point to that and highlight that. Building upon John's comments around, rare diseases like cancers, pediatric cancers, would you like to speak to how effectively other rare diseases have made trials and therapies more accessible to patients?
I think, as John said, I think that the rare cancer community has really made great strides in getting access for children with cancer, which is, we always think that people often have this idea that childhood cancer is so much more common than childhood dementia, but really it's not that much different in numbers. So I think if we can do it for childhood cancer, then we can do it for childhood dementia. And I think that a big part of that is raising awareness of the urgent unmet need in childhood dementia, and also the great potential, and about how there are effective treatments coming through the pipeline showing good results. I feel like childhood dementia has been a bit put in the too hard basket, but with the advances that have made in the recent 5 to 10 years, I think it is possible to develop effective treatments. And I think we need to raise awareness of that with the regulators, with the policymakers, governments, and research funders. And I think the collective consideration of these disorders under one umbrella will really help with that, and using that clear, understandable language of childhood dementia, which really helps with raising awareness as a collective group and get that economy of scale.
Thank you.
Thanks, Chris. I might throw to the floor now. We've got a number of questions that have come in. Maybe Matthew, if I can direct this one to you. You have covered the gamut between academic research. I know you've had some commercial interactions, and of course you're a fierce patient advocate at the moment. So the question is, a key question is to how to improve commercial viability for clinical trials for these ultra rare diseases led by small biotech. What are your suggestions?
I think the way to improve that is basically the regulatory endpoints that we can get developed for our small biotechs. They're not going to be able to have a complex behavioral instrument to evaluate children over 4 years. That's just going to bankrupt them. For the disorders that have some kind of soluble biomarker primary toxin, using that as a surrogate endpoint in the terminology of the US FDA, reasonably likely to predict benefit, that is going to be the ticket, so to speak. And as was mentioned earlier, we gathered with some of your colleagues from South Australia, colleagues from Britain and the US at the Reagan-Udall Foundation this February to make a case for one of those types of biomarkers. And that's an example in one group of diseases, but the approach could be applied to a number of childhood dementias. And we know that these toxins are well conserved in biology. For example, GM2 gangliosidosis, there is 640 million years of conserved biology that connects flamingos to humans that tells us that this compound causes fatal neurodegeneration. So in some respects, biology has pointed us to the biomarker. We just need to test that biomarker's decline as a response to clinical therapy. I think that's going to be a key to shortening the development cycle, and that is the key to developing the decreasing the cost and making it more viable for companies. That's one example of a regulatory approach that could help companies see a more viable path. Furthermore, many of these disorders may have a platform approach. Orchard is using a certain approach with metachromatic leukodystrophy. They're also trialing that approach in MPS 1 and MPS 3A. So as we expand our options, we may find many of these childhood dementias fit into groups that would allow platform technology, whatever that may look like. And I've been around biopharma and biotech for long enough that there's going to be something new that comes around in 5 years that's going to change the horizon completely. So there's a lot of hope on, but it's not fast enough. So I think decreasing the burden through regulatory pathways and platforms, et cetera, are a very likely approach.
Thank you.
We have another question here from Ellie. And Felicity, I might ask you to speak to this initially and then open up to the broader panel. She states, a lack of awareness was raised as one of the reasons for research inequity or inequity in research investment. And what can patient organisations and individuals do to help? Or is it up to the health professionals and those in research to push the current research investment in the right sort of direction?
I would say it's both, actually. The Childhood Dementia Initiative, I feel, has done great things so far and is continuing to do so more so. And and I think what's amazing about it is the combined effort. So it's grouping all the conditions together and joining forces, which is partly maybe why hasn't been as much known about it, about different conditions when there's such small numbers in each patient group. When I took there was actually no one in Australia that we could be put in contact with when my daughter was diagnosed, and the most contact we had was with someone in Norway. So and things have changed a lot since then. I was just thinking last night that social media is obviously massive for patient groups with sharing of information, but that for alpha melanocytosis has really quite only happened in the last couple of years, and it would have been instilled for other conditions many years ago.
And that's, I guess, speaking to your sentiments about the combined approach and the sort of collective effort to advocate is why forums such as this is so important, bringing together those with lived experience and those, with the clinical and research expertise.
Yeah, and and even just in understanding, not how the conditions present in the person and the symptoms that I'm thinking more even from the behavioural perspectives, and it is really great to share that sort of knowledge.
Thank you.
Felicity, I might stay with you if that's okay. There's a question from Joe that's asking, what's important for families in clinical trials? What is your measure of a success of a treatment?
So I suppose, like we were given the option with the bone marrow transplant, which was supposed, was we were very fortunate that we were able to access that with the condition that Orla has, but it hasn't been a cure. So and then there's sort of nothing else really. So I mean, I suppose it's not necessarily for families always about a cure, and if research can actually help in other ways to help quality of life and to optimize that can be just as important. And things that can provide, just like that, maybe on paper a small statistical difference could actually be quite drastic in an individual living with the condition.
Matthew, you're nodding. Would you like to add something there?
Absolutely. So I think both in the research and the advocacy community, we see in some ways two populations that we want to help. One is the population where we could see a substantial benefit, and those individuals are often the future generations, the kids who will be able to get treatment preclinically. But that doesn't mean that we should stop worrying about and concerning ourselves with the benefit to patients who are living with these diseases. And there is a lot of potential by safety in numbers, so to speak. The more we have of individuals in these similar sorts of diseases to evaluate a symptomatic therapy, to make them feel better, to make them more interactive, to make them sleep more soundly, all of these sorts of symptomatic therapies could be huge for families, and it can transform them, even though there may not be a long-term benefit in terms of the cognitive aspects of their disease. But families long for any benefit that they can get. So hopefully, if there are approved therapies that work in the pre-symptomatic child, they will also have some benefit in the child with ongoing symptomatology. But there may also be just disease symptoms that will require their own medical approach.
Right. Thank you, audience, for sending in all these questions. We'll get to as many of them as we can in the final 10 minutes. Leslie, one for you perhaps. Could you discuss the impact of viral vectors and their limitations in the delivery of antisense oligonucleotides and various gene therapies, given most therapeutic development in the area of paediatric rare disease relies on these type of modalities? Is there a need for the development of more delivery technologies and options?
It's a great question, and I think it's probably one of the most active areas in biotechnology development, is enhancing the way these different types of genetic therapies can be delivered. This is still a relatively new area of medicine, right? These studies that I talk about, our program that started 15 years ago, that's not that long ago in the scheme of things in this space. And each of the different modalities has currently opportunities to become more widely available or more accessible to more patients. So for example, the approach that we use, which is essentially a bespoke approach where each patient's own stem cells are taken out of their body, genetically corrected at a manufacturing facility and then sent back. We're doing work to enable that to be something that can be much more scalable than it is currently at the moment, where we're treating some very small patient populations. Other approaches have their own limitations. For example, for some of the AAV approaches, certain patients are not necessarily going to be eligible for certain treatments if they have pre-existing immune responses to the vectors that are used. But that's something that multiple companies are working on, to develop new capsids and new vectors that can overcome those limitations and can potentially also deliver genetic modification to parts of the body that have been difficult to deliver to today. I think that there's a lot of work as well looking at non-viral methods of delivery as an approach that can overcome many of the limitations that exist for a variety of approaches that are currently used. At the same time, I think there's still a lot of opportunity with the approaches that we have in our hands today. And it's important that we leverage those approaches as much as possible because Matthew spoke quite a bit about the challenges with the regulators. It's not just on the clinical side. They're learning the technology alongside the scientific community. And as they get comfortable with a particular approach, that can facilitate the ability for more therapies that use that approach to come to the market potentially more quickly. And it's part of the rationale for us using our approach to develop therapies, as Matthew mentioned, for Hurler syndrome, for Sanfilippo A, and potentially other diseases as well. But I think both of these things need to be happening at the same time, maximizing the opportunity that exists with the technologies that we have today that have demonstrated the ability to generate safe and effective therapies, while also improving those technologies to deliver the next generation of therapies and make them even more broadly available.
Promising, very promising, the rate of this innovation, isn't it?
Absolutely.
Maybe, John, if I can throw a question to you. There's a question from Alison. And Alison is asking, given that we have this tsunami or wave coming of adult onset dementia, and, there needs to be quite a lot of investment in the healthcare system, in research to deal with that in the future. The question is, will that have any flow-on benefit for childhood dementia?
Yeah, that's a great and actually quite a complex question on multiple levels. But I guess one aspect that we can think about is it coming back to understanding the biology of the disorders. Whilst each genetic disorder has its own biology, there may well be common final pathways that could be involved. So, for instance, neuroinflammation may be a major factor that impacts on progression of disease. And so in terms of the adult dementia space, if there are therapeutics developed that target that particular pathway, and that's an important pathway in a pediatric dementia disorder, then there would be a great opportunity to explore that particular therapeutic in the pediatric context, initially in cell or animal-based models or whatever's going to be most appropriate, with a view to potentially flipping that therapeutic over into the pediatric space, particularly if that therapeutic has already been found to have a good safety profile and efficacy in the adult population.
All right. So we've got a sort of question, and someone who's attended has shared their own experience of having a child diagnosed with dementia, and thank you for sharing that. The comment is around, even if there isn't movement on clinical studies or treatments, what progress has been made to support families and provide palliative care. So it's not just the sort of interventions, but it's actually supporting the families. And, they've shared that just the constant administrative burden and having to self-advocate for your child, and that just the drain that can have on families. Can't it just be accepted as something in need of palliative supports as well for families? So, Chris, perhaps would you like to start on that one?
I think that Venn diagram that I showed at the end shows that, we are definitely working on the care and support side as well, because we see how important that is. And we have already gained a lot of traction since we started in 2020 by bringing together all of these conditions. It's much easier to change health systems and support systems for these children. And we're working really hard in that area to support families better and get them better coordinated and multidisciplinary care. So I think that's probably almost a topic for another whole webinar. But the government's given us funding to look at the care systems and to look at the gaps and look at solutions that will better support the families. And
that's very critical work that clearly needs more research investment.
Yeah. And that was another point I was thinking about, was that it's not just the curative research that needs to increase. We also need the research in symptom management, and that clinical research and data gathering to lift all areas of care and support for children with dementia up.
Oh, Kim?
Yeah, Tiff, do we have time for one more question, do you think? Or do we need to wrap it up?
I think we probably have time for one more.
Squeeze one in? All right. There's a question from someone from an allied health background, and they're asking whether there's any experience or research, any evidence towards our ability to maybe compensate for some of the changes that are occurring in the central nervous system in childhood dementia. Is there any evidence for being able to rehabilitate in the early stages? I can open that one up to everyone if someone would like to have a go.
I think allied health is very important in maintaining those abilities in the children and supporting them to do as much as possible. But as I just said, there's not a lot of research in that area to know what the best methods are. So that's another area that could do with more research. And also there's no very little clinical data being collected about these children. So it's very hard to look at retrospectively, you know, this child's had this kind of management plan and this child's had this management plan, which has worked better, and gather that data and analyse it in a clinical context so that we can understand the best way to manage the symptoms and support the children and their families. If anyone else wants to jump in.
I think there's a body of information on areas such as intervention with hearing impairment, etc., or autistic behaviors that absolutely demonstrates the benefit of early intervention and aggressive intervention. And I think that is the basis for many people feeling that even though we may not have a treatment that approaches the root cause of the disease, newborn screening is ethically supportable just because of these improved interventions on the lives of the children and their families. And I agree wholeheartedly with that. One of the things that we're very active in is an umbrella organization representing 12 disorders, but 8 of them of which include childhood dementias, is actual direct social work support of families. And a lot of that involves developing individual education plans and making sure that, at least in the United States, that families get everything that they feel they should through their educational system. It may not be allied health necessarily, but it can be very important in the psychosocial development of the children involved as a service just provided in the educational setting.
I'd just like to say, in my experience, I've found the same thing in what Chris was saying, that like there's needs to be more information known about the conditions and the biology to apply that to the therapies. So a lot of it's always going blindly and often having the approach of doing no harm, but not really quite understanding what you might actually be doing good for, and maybe how you could adapt it to actually do better.
All right, we are at time. I'd like to thank all our wonderful panellists for this really fascinating discussion today. And Chris, am I passing to you to close?
You can do, yes. I was just trying to get the slide up that had disappeared. If you have any further questions, feel free to email me. My email address is on the website, on the screen there. Also, go to our website to find out more information, how you can get involved. And also thank you to Illumina Corporate Foundation for supporting our webinar series. And keep in touch for the next webinar. Thanks, everyone. And thanks to our co-hosts and panelists. Couldn't have done it without you. Thanks for being there for us.
Thank you all.
Thank you.
Thank you.
Bye.
Hi, everyone.